Welcome to the presentation on metabolomics. My name is Tomasz Rijavec. I come from the group of colloid biology from the Department of Environmental Sciences, Jožef Stefan Institute. What is metabolomics? By definition, metabolomics is the scientific study of chemical processes involving intracellular and extracellular metabolites, meaning the small molecule substrates, intermediates, and final products of cell metabolism. So to put it simply, it is detecting all the metabolites that are present in the system of study. And it can help us describe the molecular phenotype of an organism. In relation to the other omics levels, which you have heard about in other lectures, we might say that this is the most relevant level, but actually each level gives specific and important information on how the organism is functioning. For example, the genomics level will give us information on the genes that are present and on the potential that the organism has. The transcriptomic level will give us information on which genes are expressed, meaning how the organism responds. And then on the proteomics level, we are already getting some information about the proteins and the active enzymes that are present in the system. But then only on the last, the metabolic level, we actually directly learn about the metabolites that are present and about the metabolic processes that are happening in the system. So what we can study in the environment, there are numerous organisms present, and it is important to know what the primary, secondary, their primary and secondary metabolisms look like, and even what kind of structural compounds the microorganisms are producing. And using this data, we can deduce what effects the organisms have on the environment. But before we start, let's first have a look at how the measurements are technically done. On the one hand, we have to take care of the resolution of this complex mixture of compounds so that we are able to detect individual ones. We can use gas chromatography or liquid chromatography to do this. And then on the other hand, we have to think about how to actually detect, identify, and quantify these individual compounds that we have resolved. For this, we can use nucleic magnetic resonance, mass spectrometry, and different types of spectroscopy. Which method is the most suitable to be used is actually quite a difficult question, and there is no superior approach which you can use for all the different types of cases. You will have to decide which method is the most suitable for your system that you are studying. So for this reason, we refer you to the numerous technical reviews that are available so that you will learn more about the overview of the method and the detailed explanation how the method actually works. And then you will be able to do a proper selection of what to use. So finally, let us have a look at what kind of metabolic analysis we can do. And we name here 3 of them, which are especially interesting for microbial studies. The first one is the simplest one. It is the metabolic screening, where we try to look at metabolic profiles as a whole and do not think about the individual compounds that are present. The second is much more complex, the targeted approach, where we actually try to identify and quantify the different compounds. And third method is maybe the most complex, bioinformatics approach, which is done across multiple omics levels. So if we look at the system as a whole, in the first case, and we are not bothered by the individual compounds that are present in the system, we can treat metabolic profiles something like fingerprints. For this reason, this allows us to compare the samples between each other more qualitatively. So we will see what are the differences, what are maybe the temporal changes in a system, and most importantly, we can say something about the increase or decrease of metabolic diversity. In the targeted analysis, the second approach, this is of course much more complex because we would like to identify each of the compounds that it is present. We want to confirm its presence or its absence, and from that we want to deduce what specific processes are happening in the system. And finally, this approach is actually the basis for this third most complex approach on the omics level, where we actually want to understand which genes are involved. We want to check how and when they are expressed into active enzymes. And finally, we want to follow the metabolic processes that are happening due to these genetic changes and responses the organism has. So with all this data at hand, we can have some insight into how an organism is actually functioning in the environment. We can try to address several questions, like how the cell is sensing the environment, how the cell is sensing other cells which are close by, and when the multicellular consortia, or let's say biofilms, are formed in the environment. Most importantly, and lastly maybe, we can ask ourselves how the cells are interacting with the substrate, meaning what they are able to degrade or what they are able to synthesize anew. Because all these answers that we can provide are applicative to problems that we find in biotechnology, medicine, pharmaceutical industry, food industry, and even agriculture. So in the end, let us have a look at a more general example how metabolomics can help us design applicative solutions using microorganisms. So for example, metabolomics can help us understand which traits might be missing in a given community that we are studying, the traits which we would like to see to be present there. It can then help us find new microbial candidates which can be added to this community. And secondly, it can help us spatially structure the cells of this community to improve the exchange of metabolites between the microbes. So then finally, once we are able to do all these changes, metabolomics can help us confirm that due to the modifications that we have made, specific metabolic processes have indeed increased and are now present, and that we have achieved our goal. And in the end, I would like to conclude with a final thought. I hope that while listening to this presentation, you are already thinking of ways how to harness and monitor the metabolic power of microbes.